Welcome. Today we are going to be looking at meiosis. So, without any further ado, let's begin. First thing that we're going to do is we are going to compare and contrast the mechanisms or the purpose for meiosis and the purpose for mitosis. And so we're going to compare and contrast why we have these two different ways for a cell to end up dividing. That's first. Then we're going to diagram the actual process of meiosis to look at the different steps that take place to see how those chromosomes separate and then division, cytokinesis, takes place. All right, let's dive in. Before we get too far, though, we need to look at some vocabulary to make sure that we understand exactly what we're talking about. The first thing that we need to understand is we need to know what a chromosome is. And a chromosome is a strand of DNA that is going to be wrapped around some proteins, and it contains your genetic information, which then leads us to genes. So genes are segments of the DNA that code for a specific trait. So they code for a specific protein, which will then control a specific trait. And then you have alleles. Alleles are the variations of each gene. So a gene might control a specific trait, but there might be a lot of different variations of that trait, and each variation is called an allele. In other words, if we were looking at a gene for hair texture, it could be that we have 3 different alleles: straight hair, curly hair, or wavy hair. Each different allele codes for a slightly different protein, which is then going to result in a slightly different aspect of that same trait. Or that trait, even though it's controlled by the same gene, might appear slightly different because of the variations of or within that gene. Moving on. There are 2 different types of cells that we really need to understand. The first is called the somatic cell. These cells make up your body. They make up your skin, they make up your eyes, they make up your muscles, they make up the different tissues that make the different organs. That is the somatic cells. The other type of cells are called gametes, and gametes are sex cells. They are for reproduction only, and they are the sperm and the egg. So you have the gametes, which is sperm and egg specifically, and then you have the rest of the cells of the body, which are somatic cells. We're almost done, all right? So we're almost through all the vocab. Next thing is homologous chromosomes. So those are chromosomes that contain the same exact genes, but they might contain different alleles. For sexually reproducing organisms like humans, we have homologous chromosomes from each of our parents. So the male parent and the female parent each donated a chromosome, or each donated a set of chromosomes. So we all have 2 of chromosome number 1, for example. Those chromosomes are homologous to each other. Each chromosome number 1 has the exact same genes, but probably has slightly different alleles, slightly different variations of each of those genes that it's contained on that chromosome. We have 2 sets of each gene because we have 2 of each chromosome that we got from our parents, all right? So because we got these different chromosomes from our parents, we are diploid. We don't have 1 set of genes, we have 2. Haploid just means a cell that contains only 1 set of those genes. So if we were to look at a chromosome, there's our strand of DNA, and it's made up of different segments, different genes, which we are illustrating here by different colors. So we have 1 chromosome right here with its different set of genes, each gene controlling it for a specific trait. Homologous chromosomes would have the exact same separation of genes, the genes being in the exact same location that control the exact same traits, but they might be slightly different. So to illustrate that, we're showing slightly different colors for that variation between the 2 genes. So even though they control the same traits, because they do it differently, they have different alleles. When a chromosome replicates, that's when we typically look at it in that X shape that you're probably very familiar with. Now remember, replicated chromosomes mean that you have two sister chromatids that are exact copies of each other. Each gene on the sister chromatids are exactly the same. There is no difference between them, and they're connected at the centromere. So just remember that homologous chromosomes are not like sister chromatids. Homologous chromosomes have the same genes, but they can have that variation because they can have different alleles, whereas sister chromatids are exact copies of each other. There is no variation. So now that we've gotten this vocab out of the way, let's compare and contrast mitosis and meiosis. The first thing that we can look at is they don't have the same purpose. The purpose of mitosis is for asexual reproduction, whereas the purpose for meiosis is for sexual reproduction. Next, the input of mitosis is one somatic cell or one body cell. It is one cell that the organism has. The input for meiosis is one diploid parent cell that is going to then be converted to something very different. Mitosis, the output is two genetically identical daughter cells. They are identical to each other, and they are identical to what was the parent cell. Now, the parent cell, after mitosis, no longer exists because it divided into the two daughter cells. On the other hand, meiosis, the output is four genetically unique, four genetically different cells. And these cells, instead of being diploid like the parent cell, are now haploid. So instead of having two sets of each gene, they now only have one. So they don't have the two sets anymore, they only have one set of each gene. Why do we have these different ways for cells to divide? Why do we have both mitosis and meiosis? What's the purpose? Why is meiosis required for sexual reproduction? Well, it's very simple. The way it works out, if we only had mitosis and we had a diploid cell and a diploid cell, and those diploid cells became gametes, and they were then to undergo fertilization and come together, then we would end up with not a diploid cell, but a tetraploid cell. This cell now doesn't have two of each gene, it now has four. And if this process were to continue, we can quickly understand that we would not be able to fit enough of the genetic material inside of a cell, because it would just continue to double and double and double and double. That's why we need meiosis, because meiosis, we go from a diploid cell and a diploid cell to now haploid gametes. So when the haploid gametes come together in fertilization, now we end up with a zygote that is diploid just like the parent cells were. So we end up with a diploid cell, and that maintains from parent to offspring a diploid nature, making sure that we stay a constant amount of genetic material in the nucleus for the organisms from generation to generation to generation to generation. It doesn't change. And that's important because you do have a limited amount of space within the nucleus. So we need to have meiosis to divide the genetic information in half, so that way when it goes into the next generation, when fertilization occurs, we don't end up with double the amount of genetic information. Meiosis takes place in two stages, Meiosis I and Meiosis II. And you should recognize these different steps because they're the same steps as what you saw in mitosis. Prophase, metaphase, anaphase, telophase. The only difference now is that instead of being prophase, it's prophase 1 or it's prophase 2. And they go in order: prophase 1, metaphase 1, anaphase 1, telophase 1, and then prophase 2, metaphase 2, anaphase 2, and telophase 2. You might notice that there was no cytokinesis in between these two, and that's because it's actually part of one of the steps, which we'll talk about in a little bit. Meiosis I, first, prophase 1. it has these basic steps that take place. One, the nuclear envelope dissolves. The nuclear envelope breaks down because, again, just like in mitosis, we need to be able to maneuver the chromosomes. And the nucleus, the envelope, would get in the way. So we need that to go away. The chromosomes, they condense, so they're not going to get all tangled up. The homologous chromosomes, this is different. The homologous chromosomes form tetrads. In mitosis, this part didn't matter because you didn't have any dealing with homologous chromosomes. But now, it makes a big difference. So homologous chromosomes are going to come together and form something called a tetrad. We'll look at that in just a couple minutes. Then, when they form that tetrad, they do something called crossing over. Again, we'll look at that when we look closely at what a tetrad is. And then the spindle apparatus forms. Again, made of microtubules because it's going to control where the homologous chromosomes, or where all the chromosomes go. So first, let's look at this. We have to have the chromosomes are going to condense, the nuclear envelope goes away, spindle apparatus forms, and now we form tetrads. This is where the homologous chromosomes are going to come together and form this special structure that we call a tetrad. We call it a tetrad because tetra means 4, and each of these structures has 4 chromatids. So when they do this whole process, the issue is they undergo something called synapsis. And synapsis is where they cross over at a spot called the chiasma, and the alleles on the homologous chromosomes swap. So now, when they separate, you can see that they are no longer exact copies of each other. None of these chromatids is exactly the same because the homologous chromosomes have swapped some genetic information. And that gives us a lot of variation. But before they separate, we need to go through metaphase 1. And that's where the homologous chromosomes are going to line up on the metaphase plate. So again, our homologous chromosomes line up right along the metaphase plate, basically in the center of the cell. That takes us to anaphase 1. And in anaphase 1, very much like what you would anticipate because you remember anaphase from mitosis, well, in anaphase 1, the homologous chromosomes get pulled apart. And now that the homologous chromosomes are pulled apart, we go to telophase 1, and that again, the spindle apparatus disintegrates, the 2 new nuclei form, the cell divides in telophase 1, all right? The chromosomes decondense, and the result is we have 2 haploid cells because they no longer have 2 sets of genetic information. They only have the 1 set. So, spindle apparatus goes away, new nuclei form, cells divide, and our chromosomes are going to decondense. And again, these are completely unique cells that are now haploid. Neither one is exactly like the other, and neither one has 2 sets of genetic information because the homologous chromosomes were pulled apart. So these are 2 haploid cells at the end of meiosis 1, which leads us to meiosis 2. And to start off meiosis 2, we go to prophase 2, where again, the nuclear envelope disintegrates, chromosomes are going to condense, and the spindle apparatus forms. So, the nuclear envelope disintegrates, and the chromosomes condense, and the spindle apparatus forms, just like what occurred in prophase of mitosis. Next, metaphase 2. Metaphase 2, our chromosomes are going to line up along the metaphase plate. They're going to line up single file because there is no homologous chromosomes anymore. To go into anaphase 2, where now the sister chromatids are going to be separated, and we are going to refer to them as daughter chromosomes. And now, remember, each of those is completely unique to each other. They are not exactly the same. So our sister chromatids separate out. Now we have our daughter chromosomes. And our daughter chromosomes are being pulled to either pole of the cell, which leads us lastly to telophase 2. And in telophase 2, new nuclei form. The chromosomes are going to decondense. The spindle apparatus is going to break down. The chromosomes are going to decondense. And again, 4 new haploid cells form. Cytokinesis takes place again during telophase 2, which leads us to this process. New nuclei form, separate out, and now we have 4 completely unique cells that are all haploid, each cell containing only one set of genetic information, and each cell containing a unique set of genetic information. No 2 cells are exactly alike. And then the chromosomes are going to decondense, which I'm not going to show here because I wanted you to be able to see the different alleles that were present on each one of our chromosomes. So for chromosome 1, this chromosome 1 is different from this chromosome number 1, and this chromosome 1, and this chromosome 1. This chromosome 2 is different from this one, and this one, and this one. So each chromosome 1 in all 4 of our cells is slightly different, and each chromosome 2 is slightly different in each one of the 4 resulting cells. And again, they're haploid because they only contain one set of genetic information. Now, this is slightly different in males and females. For females, the result of meiosis is not 4 ova, or it's not 4 eggs. It's actually only 1 mature egg, because the egg needs to have a lot of nutrients and a lot of resources. So the vast majority of each division only goes toward 1 cell, which will result in the mature ova. That's why you make 1 mature ova and 3 polar bodies. The polar bodies are the result of the unequal cellular division. They are the resulting cells that will not become the egg. Meiosis I also starts prior to birth. So for human females, meiosis I starts before they're even born. Then meiosis II actually is completed upon the mature egg and sperm coming in contact with each other. So it's very close to when fertilization occurs that meiosis II will actually complete. So there's a long space in between the process of meiosis I and meiosis II, and where it all starts and where it all ends. On the other hand, in males, meiosis produces 4 mature sperm, because the sperm are not actually a source of the resources or nutrients for the actual zygote. So they end up not having an unequal division, but sperm have an equal division all throughout meiosis. Meiosis also doesn't begin in males prior to birth, but only begins upon maturity and will continue on throughout an adult male's life, basically taking about once every 10 days for the formation or production of sperm. That's it for this time. Be awesome, stay awesome.